For today, the idea is to present the results of our Cesab group that was formed 3 years ago with Paulo Guimarães and a lot of colleagues I will present. And we will together explore the networks of fungal interactions in the neotropics. And I subtitled the field guide to mushroom networks because it was not easy to find the networks in the neotropics. And so first of all, let me present some of the interactions we're interested in. We're interested in ectomycorrhizal fungi, that are fungi interacting with plant roots. This way of life of interacting and building a symbiosis where plants exchange carbon against water and nutrients from the fungi has evolved nearly 87 times in the evolution of fungi. You may know some of the fungi from their fruiting bodies. So I will speak about fruiting bodies and specimens. Those are the visible ones, but a lot of fungi do not produce visible fructification. And what we do study in terms of interaction, those are the fungi present on the roots. So you may see here some roots of plants from Peru on the white sands. We see here 2 species co-occurring on root systems. You can see that they do interact not everywhere on the roots, on the root tips, and they do co-occur with several fungi. So in general, when we study interaction, we study community ecology of these interactions involving a lot of fungi, often invisible in soils, but interacting on roots. And so the starting point of this project comes from observation at the global scale. When we look at this symbiosis here on the map in blue, we see that this symbiosis of ectomycorrhizal fungi is in general highly diverse in boreal and temperate ecosystems. But you see there is no points in the neotropics, or at least in the Amazon parts. However, there were some taxonomists years ago. It started 100 years ago. They started observing some punctually along the white sands in the Amazon. So we knew from taxonomy that there were rare places where ectomycorrhizal fungi were observed in the neotropics, but it was always punctual and from taxonomists, not from ecologists. And then this observation on the white sands was shared with many colleagues from Brazil, Colombia, Mexico, and we decided to build this project to constitute a big data set on all our rare observation of ectomycorrhizae. So that's how the Cesab group was formed. And we decided also to deepen our knowledge on interactions by joining Paulo Guimarães for the interactions, but also Diego Vasquez from the Interaction Lab, and also Lucy Zinger and other molecular ecologists interested in interactions involving fungi. So this is a really international group for the Cesab. What I will mention also, it was also that we had a postdoc involved in the project, initially 1 and then 4 and 5. So we had a lot of young researchers, Andres, Carol, Solimari, Miguel, Bernardo. I think they are all connected and did really support this project. So first of all, I wanted to reintroduce the questions we will explore today. First of all, before this Cesab project, we already explored a bit the evolutionary, the biogeography of a major group in the neotropics. Indeed, when you go in the forest, you generally see some lactiflues, some Russulaceae. This is a family highly this is the most abundant of the ectomycorrhizal observation from the taxonomic point of view. And we gathered all this collection, what we observed. So when we track here the neotropical lineages in this phylogeny, they are in red. We see that there are several occurrences of lineages belonging to the neotropics. But we observe that there is not a major diversification, except in the Antilles, perhaps. But the most important thing is that we discovered by dating this phylogeny that we had a decrease of the speciation rate from 20 to 30 million years ago for the Russulaceae only in the neotropics. In all the temperate boreal regions, it was on the contrary a big diversification from this stage. So the first question we will ask is whether we had this lower speciation rate observed in other lineages, and the other question is how far we have different scenario of phylogenetic scenario of evolution for ectomycorrhizal lineages in the neotropics. The second question we will deal with is about the habitat preferences of these ectomycorrhizal fungi. Indeed, the specimens were often found on the white sands, but Heidi Schiman and I, when we were living in French Guiana, we observed that also on slope forests, nearly half of, or sometimes 1/4 of our collections were ectomycorrhizal fungi. So they were not mentioned by any ecologists before, but we had this blue quarter was all our collections, for example, close to Inselbergs, that are granite outcrop in the forests of French Guiana. So, and this observation was also joining the observation of Maria Alice Neves from the Caatinga, from other colleagues also in the dry forest. But the interesting thing is that we never had any species in common. So it was in general a very different species. So we suspected a high endemism, and we were wondering whether, il y a un problème? Okay, whether the biogeography and the endemism was shaping this specific association with habitat, or whether there were major habitat constraints. The third question we will deal with is about the specificity of these interactions, because indeed Paulo Guimarães was the first to reconstruct networks of interactions between fungi and plants. He was already pointing out the high specialization, at least outside of the neotropics, from interactions detected from roots. On my side, from the taxonomist, I was also describing species specifically associated with hosts in French Guiana and in Guadeloupe, in the NT. And moreover, at the CRBE, there were studies also on eDNA, so just sampling soil, but detecting a habitat specificity of ectomycorrhizal fungi. So we were wondering whether it was, in all cases, we had very specific interactions involving ectomycorrhizal fungi, or whether perhaps the habitats were shaping this specificity. So for today, those are 3 axes of the 5 axes explored by Nefineo. That's what we will discuss. And of course, we will also deal with, explain a bit the feedback from this project and invite you to submit a CESAB project. So first of all, what did we learn about the phylogenetic reconstruction of those lineages? We did this work with Miguel Romero, who is in postdoc in Mexico in the team of Roberto Garibay Origel. And we wanted to compare for all the specimens we had, not only on Russulaceae, what were the patterns of fungi, especially of the endemic lineages we detected for ectomycorrhizal fungi in the neotropics. We wanted to explore first what was the microdiversity, that would be, for example, the number of sequences per species. So how far we had, it was not from microsatellite, but it was from the sequence. The sequences we had, how far we had a diversity within a species. We wanted also to explore within a phylogeny how far species were related, or whether the species from the neotropics were dispersed in this phylogenetic tree. So it was about the genetic diversity. And then we wanted to explore the phylogenetic distance between species from the neotropics, whether the species from the neotropics here in red were isolated, or whether they were dispersed in the tree. So those are the 3 aspects, and our idea was to detect how far we had similar pattern and identify some lineages that would have radiated in the neotropics. The first difficulties challenge we faced was the diversity of sources for the to gather. And indeed, so if you're interested in a similar approach for another taxonomy group, I think it would be the same challenge. So part of the data were in bold. For example, in Brazil, there is a lot of data of specimen in bold. In Unite, on the contrary, it's from Argentina. So we had very different sources of information. A lot of this information missed the geographic coordinates, and moreover, there is a lot of specimens sequenced from herbaria that are deposited in the USA, for example, and do not have the specific location from Colombia or from Bolivia. So we had a diversity of sources, but still we managed to make a huge data set of more than 250,000 sequences worldwide, gathering all sequences we had from ectomycorrhizae. And from the neotropics, okay, there is much less, because there is only like 3,500, so much less, but still there. And now some results, some important results. So first of all, we had 38 countries, islands, where we could have some specimens. So we still miss, for example, specimens from Bolivia, from Paraguay. So in case you're interested in sampling there, there is nearly no knowledge of ectomycorrhizal fungi there. There are some pictures, but no sequences. And second, we did realize that there was a huge diversity, because when we gathered all those sequences aggregated at a 97% similarity level, we still had 1,800 nearly species for ectomycorrhizal fungi detected from this dataset. So that was huge. It means there is a lot of things that are deposited in the herbaria that are perhaps pulled at the genus level, but there is a huge diversity. It's still less than worldwide. For sure, there is less diversity in the neotropics for ectomycorrhizal lineages than elsewhere, but still we detected 80 of the 87 lineages, and we detected a high diversity, more than we expected. Now, looking at the genetic microdiversity, that's true that we expected that at least some endemic species from the neotropics would show a high diversity within species. We did not detect this pattern, so it means we generally had a few endemic species that had only a few, 1 to 3 variants of sequences. So it means maybe we have a undersampling for this species, but still we detected the only difference was for the Clavulina genus. So it's a genus that was already supposed to be originated from the neotropics, and indeed we observed a higher diversity of the species endemic from the neotropics than the species from the non-neotropics, so in gray here. The second point, now on the nucleotide distance and the comparison between endemic species and non-endemic species at the scale of the neotropics and elsewhere. Indeed, non, this is pas là. Okay, sorry, it was in the room. So we detected that indeed only 22 of the lineages had a higher genetic distance within species in the neotropics than elsewhere. So it means the pattern in general is to have a lowered genetic distance within species in the neotropics than elsewhere. It's okay. But then there are some clades that had distinct patterns, for example, for Elaphomyces, from Coltricia, from Philoporia. We had for those clades, the endemic representative in the neotropics had a higher genetic distance than elsewhere. And for Cantarellus, again, Elaphomyces and Phaeocolibia, that's true that within the neotropics, their endemic representative and the non-endemic had a major differences. What the consequences of this? Indeed, it means in the phylogeny, for example, there are some lineages that show really a difference in the position of the phylogeny in the phylogeny, and also a clustering of their representatives. So, for example, that's really visible in the Cantarellus. So, in Cantarellus, here you have all this gray cluster. Here you have the position, the belonging to the neotropics. They all clustered together. And on the opposite, you have, for example, Clavaria delphus that has nearly no patterns of clustering in the neotropics, and the hypothesis that these later lineages, for example, they may have colonized several times the neotropics. So to summarize these results, it means that for only some lineages, Elaphomyces, Clavulina, Cantarellus, Phaeocolibia, we may have a long-term isolation in the neotropics, but not for all the others. So mostly it's a pattern of recent colonization. Last point on this exploration of phylogenies, about the phylogenetic diversity and the divergences. That's true that we had only few groups, only 4 major groups has really a difference in the position of the neotropical sequences compared to others. So it's Elaphomyces again, Ramaria goteria and Pisolitus had differences at major differences. In general, there is only few clades that showed really a high divergence within their phylogenies. So it means maybe we did not have so many lineages rooting in the neotropics, but that's true that we still have some higher distance for endemic, at least for Cortinarius and Clavulina. So that those may have radiated, but really, they do suggest an ancient diversification for those, but only those two among 80. To summarize this part, so we detected we can cluster and detect 5 patterns of colonization and radiation in the neotropics. We have the more common case, it's a rapid colonization and an expansion in the neotropics. So that would be, for example, the Tomentela, the Inocibe, they just arrived and speciated recently. We have a little group of older colonization events, so where we could find the Coltricia, some clades like this, but they did not really lead to radiation. So we have a lot of lineages that never radiated. We had just a few groups that show an ongoing diversification. So in particular in the Ramaria, the Tulasnella group also. Tulasnella seems really interesting there. We have the case where there is a bit of older scenario. That's the Rucinaceae. We started with a more complex. Indeed, there is both old and recent colonization. And we have only few cases, the group 5, where we have some diversification. So that's Lectifluus, the Pisolitus scleroderma, so just Entoloma and Cortinarius. So 5 groups, perhaps in other places in the world that would be also the case, to have so many scenarii. But indeed, we're happy to show that even if those ectomycorrhizal fungi are really rare, they do have a really complex biogeographic history that started years ago, the story in the temperate region. So to conclude on this part, and then I will let also Carol speak soon. Sorry. What we suggested also is that there is also a recent diversification ongoing, especially for Pisolitus, for example, for some clades that include some invasive species. So we discussed this last year with the Symposium of the New Phytologists in Campinas, in the White Sands, also in Brazil. So there is an ongoing diversification today and a presence of ectomycorrhizal fungi in the neotropics, while indeed initially we did not have this picture for this symbiosis in the Amazon, for example. So, however, to really link those phylogenetic patterns with perhaps some habitat question and the evolution and the biogeography of the white sands, we missed the habitat of the specimens, and that's why we shifted to the eDNA data, where we have much more habitat visited in the neotropics. So for the second part of the Nefineo, we did not work anymore on specimens, but we worked on eDNA to be able to see in which habitat there was some more specific symbiosis, for example. And we started our project in 2022, where there were few data, for example, in the center of Brazil and so on. In 2026, now the global fungi is really database is increasing the number of data. And thanks to the funding from Spoon, we had the opportunity to sample in Brazil, Colombia, Mexico, Argentina, in many places. So we did also added a lot of points thanks to Nefineo and side projects. We faced a major problem, and I just wanted to present these 2 slides because it's not finished, but I think it's interesting for everybody. We face the problem of having a diversity of barcodes to study those different data sets and published data sets and so on. So thanks to Bernardo Aguila, who is in postdoc in Mexico too, we designed, and Romain Frela, with their connected, we designed a new pipeline to gather data from different sources that are all including part of the ITS, which is a barcode commonly used for fungi. And the idea is to compare it to a larger database that comes from genomes to be able to make a unique assignation, a parallel analysis of the different datasets. So Bernardo, sorry, I won't discuss much more, but the idea is that it's in a publication in PrEP. I think it will be useful to a lot of other models, not only fungi. And so we are still exploring, but what we could see is that when we we are designing some parameters, especially to be able to compare different datasets that were produced, one in Mexico with ITS1, one in Brazil from ITS2. In general, we see that, we see that we may have for example, in a simple NMDS, we may have different datasets with absolutely no common points. But when we play with this new pipeline designed by Bernardo, we are trying to overlap at least at the genus level. And we see that then we could compare the different datasets. So we are still in the dataset harmonization, but it should be submitted soon. Anyway, to introduce Carol, I would say that even if we gather now, we have 1,062 sites in the neotropics, even if we gather more than 300,000 species as specimens, we still observe that ectomycorrhizal fungi are rare in the neotropics. And one question that emerged, and that's what Carol will explore, is how far those rare ectomycorrhizal fungi are important for neotropical forests. So Carol, you tell me when sending the slides.
Perfect. Thanks, Melanie. I have a doubt. How can I pass the slides?
I will do it. I'll do it.
Okay. Okay, great. Hi, everyone. It's a pleasure for me to be here to talk to you a bit about this part of the project. Thanks, Melanie, for showing these amazing advances. So my name is Caroline, and I'm an ecologist working on these species interactions and also using ecological networks. And I'm going to show you the part of the project where we are exploring the interactions between ectomycorrhizae in the roots of the plants and how these interactions shape network structure below ground. So what we did is that we took advantage of this data that was collected in the Nohag Nature Reserve in French Guiana. And we have these 6 plots there that we show in this picture. So at the Sauberg, at the Plot Lec, the Monte, the Terrace, and the Petit Plateau and the Grand Plateau. And these areas we have 9 plant hosts, 9 plant species that we show here. And we have for these plants, the roots were collected from 104 individual plants. And from these root samples, we identified their associated ectomycorrhiza fungi. Can go for the next. And we then build these interaction networks. So you can see in this network here, you have the host plants in green and the ECM in orange, but also these other functional groups. So because in this data, we could also identify fungi that are not ectomycorrhiza, we classified them in the functional groups. So we can see how they are also associated to these plants. And our main goal was to understand the variation of specialization and also the interaction structure of these networks. So what we did was that we were interested in asking if specialization varies for fungi and also for the plants. And to address this first question, we calculated the specialization index D prime. And we did this both for ECM and for host plants. So to see if there is some asymmetry in the specialization degrees. And also to check how ECM ectomycorrhizae can structure these networks, we run simulations. So basically, we systematically removed a different functional group and to test how removing functional groups change the network structure. So with this, we could calculate some network metrics. Specifically, what I'm showing today is the effects of removing functional groups on connectance and nestedness. And we chose these metrics because connectance shows how densely connected the network is. So it's a measure of the realized interactions of all possible interactions in the network. And the nestedness also shows a bit of how the network is organized in a hierarchical way, how specialists connect to a subset of generalists. Okay, now we can go. So we found that specialization is not uniform, so it varies both for when looking for ectomycorrhiza, we can see that it varies for the same species among sites and also among species, no? And so even within the same forest, we have some ectomycorrhiza that are more specialized than the others. And also we found that this also varies for plants. So there are some plants that associated to more ectomycorrhiza than the others. And when we run the simulations to assess the effects of functional groups on the network connectance, what is very interesting here is that we found, maybe you could focus on the graph on the right, that we can see the change, so the delta connectance, how removing each functional group changed the connectance of the original network. And we can see that removing ectomycorrhiza decreases a lot more than the other functional groups, the connectance. So this suggests that ECM are indeed the keystone mutualists, and that the absence of this group can have a major effect on connectance, maybe turning the network more fragmented. And the same effect we found when we were looking at the network nestedness. So removing ectomycorrhiza also had the strongest effects on network nestedness. So when this group is removed, then the organization breaks down a bit, and it may have an important effect on network stability and also resilience. And what is also interesting is that when we look at the different sites in the same forest, so now looking at the effects of removing ectomycorrhiza from these different 6 plots, can go, we found pretty much the same effect across these local networks, except for the Grand Plateau and the Petit Plateau, where removing ectomycorrhiza had weakest effects. And it may be related to the fact that these areas have more nutrient availability, for example, than the Inselberg areas and around Inselberg. And the same for nestedness, which is the next slide. We found also that, well, we see that removing ectomycorrhiza had this very strong effect at Plot-Lec and Monte and Terrasse, whereas in the other side it had a weakest effects. So, well, putting these results together, it suggests that ectomycorrhiza are keystone mutualists and that the presence maintains network cohesion. And this has important implications because if we think that their loss, for example, due to some environmental change, may lead to cascading effects, and it can affect also other fungi and the resilience of these below ground mutualist key interactions, and because their effect can propagate through the entire network. And the next steps now, we are also using some data from Julieta, that she has some interesting data from before and after a hurricane in Mexico. So we intend to assess how disturbance, in the case of extreme climatic events, can affect these networks. And now I'm going to pass to Paulo Guimarães, miúdo, please. Thanks.
Thank you, Thank you, Carol. Thank you all for coming. I will try to put in a different perspective what Carol and Melanie just said. We focus on essentially focusing on the network structure aspects. So if we shift gears here and look for this project in terms of the contribution to network ecology, we see that our study may allow us to rethink some ideas that have been developed in the past decades of ecological network literature that focus essentially on a subset of ecological interactions. So network ecology classically studies the structure and dynamics of ecological interactions in terms of the patterns of direct and indirect pathways that emerge due to the fact that no pairwise interaction are isolated in nature. And this can be done at different scales, space, time, or levels of organization. You can study individual-based networks, so how individuals share resources in a given site, to up to how species interact in a given community or even across broad spatial and temporal scales. Next, please. And traditionally, ecological networks have been focused on a small subset of the ecological interactions. It starts with the studies of food web, since the classical work by Joel Cohen, or even earlier by Elton and others. Then more recently, in the past 20 plus years, we have been studying a lot of plant animal mutualism, especially pollination and seed dispersal networks, then parasite host networks. Some individual predation has been particularly well studied at the individual level. But if you go to the literature, there is a missing component, which would be like, I don't know if the final frontier, but at least the next frontier, that are the networks of fungal interactions. Next, please. So, at my point of view, one very interesting aspect of the work, that the outcome of the work of our collaboration in the past years is try to rethink the results of some questions that we have been exploring with plant animal mutualisms and trophic interactions in ecological communities. I would say that ecological network science, and network ecology in particular, network science in general, has only two questions. How does dynamics affect structure? How does structure affect dynamics? So essentially, we can use networks to make hypotheses about the assembly and disassembly of ecological systems. So this will be how does dynamics affect structure? Or we can think if you have a particular network structure, how cascading effects will propagate through the system through indirect effects. Next, please. And we have been studying that as different groups of research groups by combining natural history information, tools from network science that come from physics, social sciences, molecular biology, ecology, and mathematical modeling. So using these tools, we can explore those two questions in different ways. Next, please. And by doing so, we can create a crosstalk between fungal ecology and network ecology. So for fungal ecology, I think that our project creates the opportunity to start to think on how indirect effects that involve species that are not interacting directly with each other, may trigger ecological and evolutionary changes in the assemblages of fungi and their hosts in a given ecological community. So how these effects propagate, at least partially, will depend on the architecture of those networks that emerge and that we are just start to describe. Next, please. But going back to network ecology, and this is something that I'm particularly interested on, is we can test predictions or of hypotheses that were developed to other ecological systems. For example, this is just one example. We have been suggesting in the literature that the degree of association between two individual partners at the individual level of different species, have effects in terms of evolutionary dynamics that will lead at the community level to structures, network structures that are quite different. If there is an interaction, so a
Le
given
réseau ici, c'est quelque chose.
sorry. A given host is individual harbors a given symbiont or herbivore or mutualistic partner, and this association is too intimate. The network, whoops. Sorry, I think that
Oh, maybe we lost Melanie.
I think so. I can try to.
Okay.
To
She
continue.
sent me the slides, so let me see if I can.
The thing that I can, if I can
Is she
try
back?
to.
Hop.
She's back. Okay,
she's back.
good. Did you lose the connection, Melanie?
Tac, tac. Non, attends. C'est le Wi-Fi qui a planté là. Pourquoi ça veut pas? Ah oui, du coup, ça m'a déconnecté de mon. On va y arriver. Hop. Pardon. Là. Sorry.
Okay, very good.
No problem. Just one slide back, please, Melanie. Thank you so much. So one hypothesis that we have in literature, that something that we have been trying to argue in the last 20 years, no, not so much, 15 years, it's that the degree of association of individuals of partners at the individual level, so like a gall-making insect living inside a host plant or an ant colony living inside a given plant, led to, at the scale of the community, to a particular network structures that will be this fragmented network that we see in the upper part of the panel. Whereas the other interactions in which one individual interacts with multiple partners across their lives, like predators feeding on different prey, or frugivores feeding on different fruiting plants, will lead to a connected network. So this is one hypothesis that we have in literature. And next, please. And by studying the fungal networks together with Melanie, Carol, and all these amazing people, I'm glad to be wrong. So actually, the data on fungi interactions goes against our predictions. So we have here the situation in which one individual is linked to a given host, a small set of partners, and at the community level, at the site level, what we have is a network that is connected. So we need to rethink some ideas on what is governing the structure of ecological networks, or at least the results for plant-animal mutualisms are not so general. Plant-animal mutualisms and predator-prey interactions are not so general. Next, please. And when now shift the attention to another aspect of the project, I would like to talk briefly about professional development in our group. This collaboration that involves several amazing researchers from different countries had a huge impact on many young scientists that were hosted in my lab at the time. So here is just a small subset of them. Most of them already left the lab, and all of them were usually impacted by the collaboration that Nefineo allowed us to build with Melanie, Diego, Roberto, and many others. So it was a real pleasure to have you guys allowing this personal growth. Some of them are now overseas, others are now, like Erika is now a professor here in Brazil. And this other dimension of the project, I think that will have a long-lasting impact in science, not only here in Brazil, but in Latin America and beyond. Now I will give the word to Melanie.
Well, thanks, Paolo. So just to conclude, we had just a few slides and we'll be short, but well, you saw that we explored the phylogenetic history of 80 lineages, so that was a first step in the neotropics. We are gathering a lot of data from observation and eDNA, and now we exploring new hypotheses on the importance of indirect interaction and from networks ecology, so a lot of inputs, but everything was done with a collective enthusiasm that we stimulated thanks to meetings, and so thanks to the CESAB. And what I wanted to also to mention is that there is a lot of tools from the cooperation between France and other countries that were used for example, fund the stay of some students longer than the CESAB group and so on. So I wanted also to mention those tools for the community of the CESAB. So in case you organize a CESAB meeting, you may contact Campus France, Climat Sud, or perhaps think about the International Research Lab. I do belong to one still in Argentina. Those labs, they do know how to cooperate and find fundings for your students. So that's something I wanted to mention. The second thing I wanted to mention is that there is also a huge project that was that exploded like our CESAB project, but gathering much more people in the world. So the SPOON is a Society for the Protection of Underground Networks. They are now gathering 88,000 points of observation around the world. And one of the major results last year was to show that there is indeed a higher diversity, at least in some places of the neotropics, that was completely underestimated. So indeed, there is much more ectomycorrhizae to discover, at least on the coastal habitat in Brazil. And finally, if you want to learn more about ectomycorrhizae, we do organize the Congress. We are part of the Journée francophone des mycorhizes, so to be held in Toulouse. It's free. You're still welcome to submit an abstract. So it's in Toulouse next June. We do organize also in South America the Congress in Quito about the mycorrhizal symbiosis in the neotropics, and it's with the support of the IRD. And then we have a training school in the Amazon every year in Peru, in the White Sands, Metamazon, still funded by the IRD. And finally, if you're not convinced that ectomycorrhizae are important in the neotropics, please have a look in your forest around in the temperate region. There are plenty of articles that already show that ectomycorrhizal fungi and their diversity represent a tipping point for a temperate forest. There are plenty of examples. Carol was mentioning the response to hurricanes. So there is a lot of cases where ectomycorrhizae are important. That's not everywhere, of course, but please have a look. And if you're interested in, we do have a project, BOSPHOR, where we will deal with this question of resilience of networks interaction in French temperate forest for the next 4 years. So thanks a lot to everybody. I'm done for this talk, trying to gather a lot of the different projects. I thank a lot of colleagues that interacted with us, a lot of colleagues that also were interested and just contributed with data. And that's already a first step to be interested in ectomycorrhizal fungi and their networks. Thanks a lot.
And thank you very much to all three of you. That was a lovely talk, super interesting, and apparently it opened lots of perspectives, and this is exactly what we hope for any research project. So thanks very much. We have some time for questions, so please, anybody from the audience, I think now we will allow you to put your camera on and raise your hands. And turn on your mics if you want to ask any questions to Melanie, Paolo, or Carlini. And if you're not able to talk, you can also just ask your questions in the chat. And also, Melanie is in a room which has people in it, so I don't know how you want to manage questions here, but yeah, just maybe if somebody asks a question in the room, you repeat it so that everybody online can understand it. Yeah, okay, awesome. So, any questions?
I have a question in the room eventually. Christophe? Moreover, come.
Close to the microphone. Hello, thank you very much for this nice presentation and talk. And I have a general question about what was presented by Caroline and also Paulo. It's about the fact that you describe different trophic mode of fungi. And I'm wondering, I was surprised not to see endophytic fungi, I have to say. And I'm wondering how this classification is so robust.
Oh. Carol, I don't know if you want to answer, but because it's me that did the trophic assignation, perhaps.
I actually could not hear well. If you can repeat to me.
Indeed, it's a good question from Christophe, Carol. So it's a question for both of us because I did the trophic assignation, and that's true that Christophe in the room here is interested in endophytes. And that's true that it was not represented, for example, in the trophic groups you had. It was indeed included in symbiotroph, not ectomycorrhizae, and they are important, especially in the Petit Plateau. And so the classical terra firme forest, they are important there, but they do not appear as structuring for the networks on the granite outcrop and low canopy forest.
Thank you.
Other questions?
I had a question, but it's really from somebody who doesn't know anything about fungi.
Okay, we have it.
So
okay.
you have one in the room. If there was in the room, I'm happy to.
Go ahead, Camille. We'll be back in the room. Go ahead, Camille.
I don't know. I was just surprised to learn again, or maybe it's something that I learned wrongly when I was in school at uni, that I thought that the neotropics were actually quite rich, in mycorrhizal species. And I thought that was even part of the theory of how so many species could actually live in the same amount of space. But yeah, so I don't know. This is like the very beginning of your work. Actually, I was like, really? I had no idea.
In general, what we learn is that for fungi in general, that's true that the neotropics are highly diverse, especially for endophytic fungi, for example. But ectomycorrhizal fungi were one of the only groups that was more diverse in temperate and boreal biomes. So it was the general way it was taught. For endomycorrhizal fungi, that's true that there are a diversity of mycorrhizal symbiosis. For endomycorrhizal fungi, they are abundant in the neotropics, but they are not highly diverse because there is not such a high diversity anywhere in the world. It's a different way of measuring also the species diversity, but they are not more diverse in the neotropics. So, but I would be curious to look at what course you had. Perhaps it was just the general pattern for fungi in general, but for endophytic fungi, that's true, they are more diverse there.
Maybe, yeah. I don't know. Okay.
Yeah, but that's true that, for example, for people working in forests, in temperate forests, in general, they are used to know boletes, russules, chanterelles, and so on. But in the neotropics, not at all. But they are present, but it's just endemic species and really rare. Géraud? Là, on peut t'entendre.
So I have two questions. I will start with the first one for Paulo. Your hypothesis about interaction intimacy, you said that it does not apply to the fungal networks, but I'm wondering if it's not because you are studying different trophic groups within fungi, if I understood well, and while the other networks you were presenting between ant plants or birds and fruits were related to only one trophic level or one functional level.
That's is a very good hypothesis. Although when we analyze different groups in isolation, usually those networks are not as fragmented as the ant-plant mutualisms, ant-myrmecophyte mutualisms. those are very fragmented networks, or the interactions between plants and gall-making insects or mining-making insects. They are very, very fragmented. The same is true for seed predations like endophagus, predispersal seed predators. They usually, like insects, they are very fragmented. So I think that you are right that the combination of different functional groups will allow to create links that would among groups that will otherwise be isolated. But even when we do that in some of our analysis, the degree of isolation of the groups in the network, what we call network components, the number of those components and the size of them are more close to connected networks than to the highly fragmented networks that you observe in some plant-herbivore interactions.
Okay, and you had a second question?
Yes, if I can have a second one for you, Melanie. I'm wondering let me think what I wanted to say. You were speaking about endemicity and diversity of ectomycorrhizal fungi, and I'm wondering if you had a look at the pattern of these fungi and the plants across the neotropics, and if you can parallel the endemicity and diversity because they are plant specialists.
Well, that's a good question. Indeed, that's the limitation with fruiting bodies. When you sample the fruiting bodies, you do not know the host necessarily to which they are associated with. And in general, they grow in forests where there could be 200 different hosts. That's a question we could answer with the root tips, but we do not have a huge database on root tips. But root tips would be the way to really answer this kind of question, because we can identify the plants from the root tips. But from the fruiting bodies, you sample it, but you do not know the plants. And from the eDNA and soils, that's the same thing. We could make parallel, knowing the host, in some places. And indeed, we did both contribute in the article of Heidi, where we saw, for example, correlation between the matrix, the tree diversity and the community, the changes in fungal communities, but we cannot go further.
Do you mean that you use not only ectomycorrhizae as roots with secondary, but also other sources of biological material?
Well, perhaps it was not clear enough. I tried to put some pictures. The first part was on fungi, on the fruiting bodies, the specimens. The second part was on eDNA, and was just the method. And the third part was on root tips.
Okay. But I guess that you have many fruit bodies that are not from ectomycorrhizae.
No, it was only fruiting bodies from ectomycorrhizal lineages that we studied in the first part. Yeah. So for all of you, that's true that ectomycorrhizal habit is nearly conserved for some genera. So for some specimens, we did not have the species level assignation, but it was sufficient to attribute them to lineages of ectomycorrhizal fungi. But that's true. That was one of the challenges in interactions. We may work only on the fungi sometime, on the fungi and the plant sometime, and sometimes just on the visible part of the fungi. So sorry, if it was not clear. Other questions? Well, if some are interested, we have a postdoc offer, which is in France. So indeed, I have two things I wanted to share. First, with Antoine Brun, we have a postdoc offer funded by the OFB to work on the indicators we want to evaluate about the resilience of forest face to climate change. So it would be more to accompany the monitoring of French forests that are let in free evolution, but through perhaps indicators based on networks. So if there are people interested in networks, that would be interested. And the second thing is that I think we will apply to a CESAB after to make more links between what we've learned from the neotropics and what links we could make with the temperate forest. about questions typically about networks resilience and so on. So if there are curious people about networks and parallel between different ecosystems, you're welcome.
Awesome. Well, thank you very much. Then I think that we can call this a session. And so thank you everybody for assisting. Thank you to our speakers for the talk. It was really interesting.